Okay, now let's have a class. So we're gonna go over the results, and then also today's major work is two of you gonna work together to do the gram stem of 2 colonies on your strict plating plates and finish the lab report of the Unknown-1. But the results is clear, which is E. coli and Serratia. And so we want to talk about something first. The results from last week, from Tuesday, you should see the first one is your glass tube. Okay, this is what you have, a glass tube. That is a king cap tube. We inoculate Bacillus subtilis, and we incubate it 35 degrees Celsius for 24 hours. Okay, what you see it? You look at your own tube. Did you see any of these looks like turbidity? If you see it looks like turbidity, which means the bacteria is growing there. And then you look at the bottom. Did you see the precipitation? When you see the precipitation at the bottom, which means it's also growth. So these are the 2 markers for the bacteria growing. Actually, there is 3 markers. You can see 2 here, so we want to mention. Okay, the 3 markers for bacterial growth. Number 1 is you will see the whole tube become, we say, cloudy or turbidity. And we will mention in the lecture how cloudy, how turbidity it is. We could use a spectrometer to do the testing to see the OD value. But the only thing we cannot determine is whether the bacterial cell is survived, they're dead or alive. But you do see if the bacterial broth after incubation, it looks like very cloudy and the turbidity, which means the bacteria is growing. Second, you see lots of things at the bottom. So this is second marker is precipitation. If you see the bacteria, lots of them at the bottom, we call precipitation. That means bacteria also is growing. Second, last one. Sometimes for very strict aerobic bacteria, they will be stay at the top. That's called the pedicus. Will be stay on the top. So those are the 3 markers for bacteria to grow. This is very important if some of you are nursing students or in the future you're going to work in the hospital. When you change a glucose or saline solution bag for the patients, when you took them out from the refrigerator, you see the glucose bag is already see the turbidity or cloudy or precipitation. That means the bacteria is already growing, which means you cannot use it. It's already contaminated or expired. Okay, so that's important information. Same for the physiological saline. If you see the physiological saline bag in the hospital, you took it from the refrigerator, you see those 3 markers, which means they are already contaminated with microorganisms. So this is the first one. Second one, slant. So everybody look at your slants. What it looks like. You should see a very clear zigzag marker. You look at your slants. If you see a very clear zigzag marker, which means you did a good job. And this is for temporary storage bacteria at like 4 degrees Celsius for about 7 days. 4 to 7 days, less than a week. Okay, so that's easy. That's a big deal. Did you get your good results on your spread plating. Oh, sorry, streak plating. I think if you did not get a good results, I have a 4 very good example myself. You can take one. This is the one. This is the second one. You can see a good colony there. That's another one, and that's the other one. And we can go over the results. When you here, you see there is a 2 different colonies. Is that right? 2 different color. One is red, pink. Another one is colorless. These are the 2 bacteria we're gonna pick to do the gram stain. And you tell me what the gram results will be looks like. Okay? So that's the first thing first. Today's work we'll do unknown. So you already did is a streak plating. We at 35 degrees Celsius for 48 hours. And you will see 2 different colonies. One of them are very red. And first of all, we're going to do is colony morphology description. Where it is. On the front of each desk, I printed out one of the pages on your lecture in the color. You can use as a reference, but please either take a picture or return to the front of the bench. I only have 20 color printers. I can only print it out 20. So this is you use as a reference. What you're gonna see, is that gonna be circular, filaments, irregular, spinal? That's for the form. For the elevation, is that going to be flat, raised, convex? For the margin, it's entire, undulate, locate, erode. You could be compared with these different morphology. And be careful, this is important information, because some of the bacteria, you will get like a pushable colony. For example, Neisseria gonorrhea or Neisseria meningitis. When they grow on the chocolate agar, it's like a pushable colony. So the colony morphology is also important. But there is something I also want to remind you. When you did on the slides, this is the cell morphology underneath microscope. What we are talking about today is the colony on the plates. Those are different. Okay? This is the first thing. The second thing, you have a pick colorless colony. And then you pick red colony. And both of these we will do gram stain. This is the same thing we already did. So you should all familiar with the procedure, including crystal, violet. Second procedure is gramidine. Third one, 95% alcohol decoloration. And the last one is counterstain with saffroning. And I want everybody do by themselves. Then 2 of you work together, pick the good results. Okay? So you should have 2 bacteria, which is EC, another one is SM. EC stands for E. coli. That's easy. What is SM? That is Serratia. I need to make sure that spelling is correct. So I checked on the website. Serratia marcescens. This bacteria is very interesting. At 25 degrees Celsius, it is red color. At 37 degrees Celsius, it is colorless. Now you're going to tell me what's the morphology, arrangement, and the gram reaction. So you're going to tell me the thing is underneath 100x, of course 10x you find the specimen, and underneath 100x with oil immersion. You need to tell me three important information. Is gram positive or negative? Number two, morphology. Number three is arrangement. Okay? You need to tell me. You need to pick is a single colony, not a mixed culture. All the others are the same. So use loop, draw the circle, label. One is pick the pink one, the red one. Another one is a colorless one. And make sure you flame your loop when you prepare the smear. Don't do the cross-contamination. Okay? Everybody do by themselves. One slide, two colony. Underneath microscope, take a good picture. Two of you work together to choose the best results. Write a lab report, which is 2 to 3 pages. And the requirements is at the last one here. We will due by 2 weeks from today. Okay? This is Thursday, so 2 weeks from Thursday, you're going to turn out your first lab report, and that is 15 points. Now keep in mind, this lab report is teamwork. It is as important as your exam because it's accumulated in the whole final whole 200 points. Okay? So go ahead to do it. If you have a question, ask Rion. Rion can help.